In this video, we're going to talk about restriction endonucleases, ligases, and molecular cloning. So there in the 70s was discovered a group of enzymes from bacteria, so they're naturally endogenous to bacteria. These enzymes are referred to as restriction endonucleases, or just restriction enzymes for short. And what they do is they recognize each one, so HPA1 here, recognizes a very specific sequence of DNA, and then it cuts in a very specific place within that sequence. So these enzymes are all ways that scientists can use to cut DNA in very specific places. And again, we haven't engineered them. Scientists haven't engineered these enzymes. We find them in different species of bacteria. Now, this bacteria, what these enzymes are originally meant for is as a defense mechanism against viruses. So viruses try to infect bacteria, they inject the DNA into bacteria, and then the bacteria start making these enzymes that cut up that viral DNA. We've just taken advantage of this, purified the proteins from bacteria, and then we use them like scissors to cut DNA in very specific places. Now, one thing you'll notice is that some of the cuts, right, here's a sequence right here that's recognized by a specific restriction enzyme, but it cuts at two different places on the two strands. It cuts right here between the GNA and right here between the GNA. Also notice that all restriction enzymes recognize palindromic sequences. GAATTC on the 5' to 3' one strand, GAATTC 5' to 3' on the other strand. So they all recognize these palindromic sequences. Now, one other thing I didn't mention is that some enzymes, some of these restriction enzymes recognize 1, 2, 3, 4, 5, 6 base pair sites. Some recognize 12 base pair sites. Some recognize 4 base pair sites. And we can take advantage of that and use that to cut DNA up into either larger fragments or smaller fragments. Now, I want you to think about that before our next meeting, is what would the size of the recognition site, what effect would that have on the average length of the fragments that are generated when you incubate that DNA with the different restriction enzymes? Okay. All right, so let's get back to this. These are called sticky ends when the restriction enzymes cut in different places on the two different strands. Sticky meaning that they end up with these, the fragments that are generated each have this overhang here. And that overhang then can form hydrogen bonds with reverse complementary overhangs. They are sticky to each other. Whereas something like HPA1 has a blunt end cutter, right? It recognizes this sequence here and then cuts at the exact same place on the two strands. So that has blunt ends. It doesn't have any of those overhangs, those single-stranded overhangs anymore. Now, let's think about some advantages of this. Well, the advantage of sticky end here is that as long as you are connecting them to fragments that have been cut with the same enzyme, those ends will have the reverse complementary overhang and will then be able to stick together. So the reaction that has the two fragments sticking to each other and gluing them back together again, or gluing two different fragments together, is going to be more efficient, right? Because they will stick to each other and find each other better. However, what that means is the two fragments that you're trying to glue together must have been cut with the same restriction enzyme and therefore have the same overhangs. Now, blunt end cutters, the reaction necessary to glue two blunt ends together again, or together if they're two separate fragments that have never seen each other in the first place, is going to be less efficient. Okay, when you try to glue them together, there's no overhangs to stick together. However, as long as the enzyme produced a blunt end fragment, doesn't matter what enzymes are used, right? Because a blunt end is a blunt end regardless of what enzyme made that blunt end. And so here you can use any enzyme as long as it causes a blunt end. And so you have much more flexibility within that regards. Now, ligase. Okay, so we have restriction endonucleases are the enzymes, are molecular scissors. They allow us to cut DNA in a very specific place. But that's not just what we want to do, right? We want to recombine DNA. We want to take DNA from bacteria, and we want to take DNA from humans, and we want to recombine them. And you'll see why we want to do that in just a moment. So not only do you need the molecular scissors, you also need the molecular glue, and that's ligase. Now, ligase is a natural enzyme that exists in our cells, that exists in bacterial cells. It's an enzyme necessary to repair damaged DNA in our cells. Well, we take advantage of ligase and we use it to glue fragments of DNA that we just created with restriction enzymes. So here we've taken human DNA, we've taken bacterial DNA, we have cleaved them with an ECOR1, that's a restriction enzyme, that generates these sticky ends. Well, we're going to take one fragment here of human DNA, and we're going to take one fragment here of bacterial DNA. Notice, right, because they were cut with the same restriction enzyme, they have reverse complementary overlapping reverse complementary sticky ends. We incubate them together, we add ligase, and we allow it to then seal up those two phosphodiester bonds. The phosphodiester bond between this pink G and this blue A, and the phosphodiester bond between this pink A and this blue A here. And now you have a nice, normal fragment of DNA consisting of stuff that came from bacteria and human. But there's no way to detect the difference anymore, really. Okay, so it becomes a normal hunk of DNA that you can do something with, and we'll explain what we do with it a little bit later.